All right, welcome back to our overview of Chapter 7 of Campbell's Biology, Structure and Function of Membranes. Here we're going to move on to Concept 7.3. So passive transport is diffusion of a substance across a membrane with no energy investment. Okay, so here what we have is a membrane. This membrane has small pores in it and allows things to pass through. Here we have a bunch of dye molecules. This is all in water. And to kind of understand this is each one of these each one of these dye molecules are kind of bouncing around. Occasionally they will bounce off the membrane. Sometimes they will pass through the membrane. Over time, when you have a high concentration of dye molecules over here and none over here, you're going to slowly start to move more and more over here because there are none over here to move back. So they can either bounce off or they can go through. One of the two things is going to happen. So over time, these will go through. Now, the ones that are on the other side of the membrane are also moving independently and randomly, and they could bounce off the membrane or they could go through. But it is much more likely when you have fewer molecules over here that they'll bounce off as opposed to go through. Here, when we have lots of molecules, there's a lot more chances for it to actually go through the membrane. If you think of each molecule moving as an independent chance, having a lot of them means more are going to go through. Eventually, it's going to reach a point where the concentration is the same on both sides. So you have the same number of molecules on both sides, and then the likelihood of a molecule passing through this way and another molecule passing through the other way are kind of equally likely. This is something that we call dynamic equilibrium. Okay, so when you reach equilibrium, it's not that the molecules aren't moving, it's just you're getting the same number moving over this direction as are moving back the other direction. Now, this idea, when you use diffusion to move a substance across a biological membrane, we refer to this as passive transport because you don't need any energy to make this happen. Okay, let's take a look at when you have more than one solute. So here we have a high concentration of orange and no purple. Here we have a high concentration of purple and no orange. So the orange molecules are very likely to actually pass through because there's a lot of chances for it to move over here, but no chances for a purple to move from here to here because there are no purple, right? The same thing is true over here in the opposite direction. So the likelihood of a purple going through is high because there are no purple molecules to move back. So over time, the purple molecules will try to move to this side, and the orange molecules will try and move to the other side. As this happens, the concentrations start to change. So now you don't have 0 yellow, you just have 3 yellow, or orange, whatever we decide to call them. But this ever so slightly increases the possibility of moving back through, but still it's much more likely that one from over here will move through to here as opposed to this moving through here. Just looking at random chance of it can either bounce off or it can go through. So the opposite is going to be true for the purple. So over time, the purple is diffusing to the orange side and the orange is diffusing to the purple side. When you reach a point where you have equal number of molecules on each side, then it's equally likely that it'll either stay on this side or that you have one going through one way and another moving through the other way. So you'll always end up having the same amount on both sides. Now, one thing that we have with a biological membrane is the fact that water can pass through the membrane, and it can pass through those aquaporin uh, proteins, right? So those proteins are acting like the pores in this membrane right here. Whatever these green molecules are, they are too big to fit through the hole. So the water can move, but they cannot. This net movement of water is referred to as osmosis. So if you have a lot of these green molecules over here, but only a few over here, water will tend to move towards the side. And one way that you can think of it is the water is interacting with these green molecules. So the water molecule is coming in and hydrogen bonding. Consequently, if it's hydrogen bound to this larger molecule, it cannot pass back through. So the net movement of water will be from this side to this side so that it can hydrogen bond with these green molecules. Here's kind of a zoomed out version of that, right? So here we have liquid in a tube. So we have water in this tube. We also have sugar molecules. We have two different concentrations. So this could be like Karo syrup, and this could be like sweet tea. So it has a little bit of sugar. This has a crap ton of sugar. What's going to happen over time is these water molecules are going to pass through, and they're going to hydrogen bond to these sugar molecules that cannot pass through. And what this results in over time is more water is going to pass through to this side. And if you look at it in terms of molecules per cubic piece of space, or a cubic amount of volume, over here, it looks the same concentration as over here. When we look at it over here, clearly we have more green molecules per a given unit of volume compared to over here. So if we take a line, draw it across here, we have like 5 here, but only 2.5 over here. So we can see over time the water is going to move because it is hydrogen bonding with these green molecules. The net movement of water is going to come around. Now, that same thing will hold true for water going into and out of a cell. And different things happen depending on what kind of cells we have. Let's start off looking at a red blood cell, an animal cell, and look at a scenario where we have what's called an isotonic solution. So this is essentially the measurement of how much stuff is in the water, right? So an isotonic solution is equal on both sides. So here, the same amount of water is going to be flowing into it through those aqua and porin as are flowing out. If we take it and then put it into a hypertonic solution, so this could be in either a very thick, viscous, sugary solution or maybe into salt water, what's going to happen is most of the water is going to flow out to interact with those ions or molecules that are on the outside. That's what happens in a hypertonic solution. So the cell will become shriveled up. If you think about a hypotonic solution, that is where the solute concentration is less than inside the cell. Water is actually going to be net flowing in, and an animal cell can't take that kind of pressure, and that would actually cause an animal cell to bust open. Now, plants are a little bit different in that they have this rigid wall surrounding them. So they actually prefer to be in a hypotonic solution because they can maintain pressure within their cells, and they can actually use that pressure to drive nutrient movement throughout the plant. This actually makes sense when you think about it. If you take a flower and you cut that flower and separate it from its source of water, it is going to become flaccid. It is going to start to wilt. And that's what happens when you're in an isotonic scenario. If you move into even a worse case scenario where you're hypertonic, you can actually see that the cell membrane is shrinking up inside, so the cell is actually shrinking within its own cell wall. So this kind of paradigm helps explain why we put flowers in water and why when we have vegetables in the grocery market, we constantly spray it with water. That is pure water, and it is going to be hypotonic in nature, and it is going to keep the vegetables crisp. Now, when you take an animal organism and put it into an aqueous environment, that aqueous environment might be hypertonic or hypotonic, in which case this can cause a problem for that animal. When animals need to live in this type of environment, such as this paramecium here, it has to evolve some type of mechanism to control this. So here we're talking about a paramecium living in a freshwater lake. That is going to be a hypotonic solution, and the cell is going to be constantly in danger of bursting. What it does is it actually has a special organelle called a contractile vacuole, which utilizes actin filaments and myosin to contract and actually squeeze water back out. Okay, so it kind of actively pumps the water back out so that it doesn't burst. Now, in a biological membrane, oftentimes this passive transport is aided by proteins. So think about water and aquaporin. The water itself can't pass through the membrane, but it can pass through these specialized water channels. This is something that we call facilitated diffusion. Okay, so the proteins are facilitating the passive transport of the molecules. These transport proteins that are involved in this facilitated diffusion could be channel proteins or something called a carrier protein. So channel proteins provide a corridor to allow things to move through. A perfect example of that would be aquaporin. Okay, when it facilitates the movement of an ion through, we call that an ion channel. And there it's just moving through based upon natural diffusion, right? So there's no energy going into the system. Now, sometimes these ion channels can be gated so that we can open and close them in response to some type of stimulus. That would require some type of energy input to open or close it, but then we have a way of facilitating the movement on a controlled basis. Now, a carrier protein is something that doesn't have a full channel going through. Instead, what it can do is change shape. So here we can see it kind of opening up clam-like, and it has a little spot where it can hold this molecule inside. So the molecule will go in, fit into that space, and then it closes and then opens on the other side, allowing it to go through. Again, this does not require any active energy input because the molecules coming in and binding, it could reopen up on this side and flow back out, or it could open up on the other side and flow through. Each is equally likely, but since there's more molecules over here, the odds are is that it's going to go through, right? So, looking at concept 7.3, passive transport is diffusion of a substance across a membrane with no energy investment. This is just looking at the random chance of each individual molecule and then seeing, well, what is the total number of chances in a particular compartment. So 0 chances over here, a lot of chances over here. So consequently, the molecules are going to move into that empty space. When you get equal amounts on both sides, it's equally likely that they're going to move through in both ways. So you've reached equilibrium. That equilibrium, if can drive the movement of water across a membrane. It can lead to things such as lysing cells or causing cells to shrivel up. and is facilitated by proteins in a number of fashions.